you from Stockholm, Sweden, my morning. And I have the pleasure of presenting a solution that we just released last week as a service. And this is an analysis platform service for, as Simon mentioned, for whole genome sequencing of the SARS-CoV-2 virus and helping empower laboratories in the surveillance effort. So I'll briefly go through the solution, a little bit of the background, motivation as to why, and then hopefully we'll get some questions later on that we can address. So I don't think we need to see yet another legal disclaimer, but what I want to do is just orient you to obviously QIAGEN, and especially QIAGEN Bioinformatics, which where we're called QIAGEN Digital Insights. In fact, we're one of the 3 business areas within QIAGEN, and our strategy is actually to help, especially within the clinical market or even research markets, help scientists really elicit new insights from their data. Especially from NGS types of data, which is very complex. And additionally, I think it's important to know that QIAGEN Bioinformatics has been present in a number of years in a number of important projects, and especially within virology. So I think it establishes us as quite a credible organization within viral genomics and this type of research especially. And this is very, very important during this pandemic. And we've done a lot to address this. And so this solution I'll be presenting today is one of them, and then you'll hear a lot of other additional use cases and solutions as we go along. So why are we actually interested in surveillance? And I'm not, working obviously in public health, or I'm not an epidemiologist or even a virologist, but I think it is of common interest, in fact, and in fact, there's a daily chatter about the different variants that are sort of being seen or discovered with respect to SARS-CoV-2. This is in the media, and this is, of course, of great public interest now. And in fact, there is a very important motivation for understanding these variants, because some of them, in fact, may have clinical relevance. And that meaning that we need to understand what these so-called variants mean from a clinical perspective. Would they be, for example, causing a greater degree of virulence, or would they lead to some other host adaptation, or would they be able to escape these newly approved vaccines we're seeing? And in fact, as we go along, as we've seen with other viruses, there may be treatments coming, approved treatments that is, and we're also going to have to monitor how these viruses, even going forward in the future, are able to evade and adapt to avoid these treatment options that we hope will come as well. So there are a lot of reasons to keep track of these variants, and not all of them, of course, have a negative, let's say, clinical impact, and some we don't even know. So what we're doing and what we're hearing about is really, we're kind of watching what's going on, and we're trying to understand at the same time. And one of the ways to do this, of course, is through this surveillance effort, okay? So it's very important. And governments see this as important. Public health obviously sees this as very important. And recently, so in the past quarter, I would say up until the last parts of 2020, we're seeing also a great degree of funding being released, a lot of support and guidance as well from public health institutes and certainly funding organizations to make the surveillance effort possible. But this actually becomes somewhat of a scaling issue. So while there might be funding available, while there might be technology out there, to be able to help in this effort, it's not entirely trivial, in fact. And one thing that we're seeing concretely is that, for example, the European Center for Disease Control and Prevention is recommending about 10%, if not more, of all PCR positives are actually going towards whole genome sequencing. And if we remember back before the vaccine, we were seeing case rates of in the thousands, tens of thousands positives per day. So this leads to a massive sort of scaling problem if everyone would now need to sequence 10% of these positives. All right. So yes, it becomes a scaling problem. So it's not just problems, there's also opportunities. And this is what we're seeing. This is what we've heard from you, many in this call today, and all the interviews we've done before we actually released our solution last week. In fact, we're seeing that laboratories with extra sequencing capacity are being asked or recruited in some cases or applying to be able to help with the surveillance effort. And again, surveillance in this context really means whole genome sequencing of SARS-CoV-2. And this is usually from PCR positives. Okay. Now, the challenge in that is that obviously, if you have a laboratory that doesn't have experience but has sequencing technology, then implementing these new methods requires export resources, perhaps, or new infrastructure and certainly workflows. And this requires time, money, obviously, to be able to do this. So it's not entirely trivial. Whereas expert laboratories, you would think, like public health, they might just as well scale up. But this is also not entirely trivial. So scaling up from an existing operation is, again, a pain point. And there are a number of other opportunities and challenges associated with these that you can kind of read for yourself there. But QIAGEN takes this very seriously, and I think early on we saw that QIAGEN as a company came with a lot of opportunities to help in the diagnostics. So obviously on the PCR side, the detection side, even in the antibody side, for example. And then clearly in the research areas, so host pathogen interactions and such, which you'll hear about later on as well. But now with respect to surveillance, now we really have to think about the full characterization of this SARS-CoV-2 virus. And this is, again, through whole genome sequencing. So while we have also panels, so the QIAseq SARS-CoV-2 panel, there are also a number of other vendors out there today that are providing different technologies to be able to help sequence this. And we, within bioinformatics, need to be able to support all of these technologies going forward. So this brings us to the QIAGEN COVID-2 Insights service. And I think the key points are that this is a service that we started really with the surveillance use in mind, or the surveillance use case in mind. So we wanted to make sure that we created an agnostic solution that could provide or empower laboratories using any technology. It needs to be fast, accurate, obviously, secure, and scalable. And this is very important. So we've chosen to use Amazon Web Services platform for this. It's super secure. And I would almost say infinitely scalable, but it's also got a global footprint. So we see this as a global solution. We wanted to make sure that you didn't need advanced infrastructure or expert resources to get started. So we want you to be able to be day one ready, i.e. starting when you want to start analyzing, we want you to be able to provide the platform so that you can instantly scale up from your first opportunity. And one of the ways to do this is to completely automate the analysis. So this is what we've also implemented. And naturally, it needs to play well with other systems. So basically, this is sort of the design, some of the design requirements that we've talked about. But, importantly, because there are a lot of other systems out there, importantly, we've decided to take this very seriously. We put our global operations team on it to make sure that we are monitoring and taking care of this business critical system at all times. So if anything would happen, we're always there to take care of it, and we fully support this system. And this is very important, not the least of which, the results of these this analysis needs to also be reviewed by scientists somehow. And what we do, or what we have done, is we've decided to make some decisions with respect to reporting, which you get a glimpse of on the right-hand side here. But also we've connected this to our existing platform, the CLC Genomics Workbench, so you can easily and for free visualize the data that's being produced. So it really makes a nice, full, complete platform for anyone to be able to use. So this report is looking pretty good, and this is what we call a human readable report, but let's take a closer look at it. And in fact, this is just one of the many results generated. But what we wanted to communicate here are simple and critical piece of information, such as the lineages and clades, which are obviously some of the important things that we're tracking today, but also some key QC metrics, and of course the variant summaries, so individual variants, for example, both at majority and minority. So we're able to do both of those and present that in a single report. And I think this is very important to be able to communicate around specific samples. Now, how do you get to this data? How does this generate it? It's very simple. Basically, at your site, you have a sequencer. You will generate FASTQ files as a result of the sequencing, of course, at some scale, whether it's a mini-seq or a thermal platform or a NovaSeq. So it might be tens or it could be tens of thousands. These will generate these files. These will be automatically actually uploaded to our service using some automation capability. And then these will be massively analyzed using the service. So it'll go through a standardized pipeline, which includes, of course, the pangolin and nextclade identification steps, so the variant calling, reference mapping, et cetera, at majority and minority levels, and then even generating consensus, and of course these reports that you saw. Also including the standard formats like VCF and XML and whatnot, so that you can later share the data further if you'd like to do that. So all automated upload, analysis, and automated download. It's like a factory for genomic surveillance. And like I said before, at the end, if you want to, you can go and do a deep dive into the data itself for free using the workbench. And if you had a licensed version, you can easily do downstream analysis as well, like, for example, making some phylogenetic analysis, as we see here. So just wanted to kind of summarize after this brief presentation about the COVID-2 Insights service. So, this, again, I think is a solution that or service that we've released to be able to empower and enable laboratories that maybe aren't always or haven't thought of themselves as working within viral genomics, but want to be part of the surveillance effort. And I think that with this solution, we can help you and also help those expert labs scale up tremendously and take a lot of the pain, because time is of the essence. And with this sort of solution, we can help immediately and help with the real-time surveillance effort. So I want to leave it at that and open maybe for, I don't know if questions come now, Simon, or afterwards. But yeah, thank you very much for your time and attention.